Hello lovelies, in this video the brilliant Dr. Edwards is going to be going through gene linkage for your A-level biology. Now she's going to be taking you through lots of examples here, and there are lots of new keywords to come up. So that is one thing I really want you to pay attention to in this video, writing down correct definitions of the keywords.
Gene linkage. There's two types, sex linkage and autosomal linkage, and we'll look at both of them. And these are both ways that can change the expected ratios that we'd be expecting from crosses. So quickly recap about sex chromosomes. We should have hopefully done this at GCSE. So in mammals, we have two sex chromosomes. We have the X and the Y chromosome, and that determines biological sex. In humans and in other mammals, males only have one of each chromosome. They have an X and a Y, and females usually have two copies of the X chromosome. Now, disclaimer alert, so obviously other chromosome patterns exist in nature, especially in mammals, so XXY exists, but these are quite rare. And other organisms use different letter combinations as well. So you might see in the exam there might be other letters used, other combinations, three sex chromosomes, for example, you never know. Punnett squares for sex inheritance are quite easy because it's always going to come out that it's 50% chance of either getting a male or a female. Eggs can only give X chromosomes, sperm, it can give X or a Y chromosome. And the ratio is always 1:1. So the only thing that kind of maybe is extra that may you not have seen at GCSE is you could be asked as like a supplementary question if you're asked to calculate a ratio or a prediction for an outcome, so the probability of getting a certain characteristic in an offspring, but you might be asked to specifically look at getting a male with that characteristic or a female with that characteristic. So for example, what's the probability of getting a blue-eyed girl or a girl that has this genetic disorder? So basically you need to work out the probability of getting the child with that characteristic first, and then you can just divide it in half because that's the probability of having a boy or a girl. That is if we're not talking about a sex-linked disorder, which we'll look at in a second, and you'll see why that is slightly a bit more complicated. But that's kind of a recap of what we should hopefully be expecting to know from GCSE, and we need all of that information to be able to understand sex linkage. Okay, so sex-linked genes are genes for specific characteristics that are found only on the sex chromosomes. And so they're referred to as sex-linked genes because genes for these characteristics can only be inherited on the sex chromosomes, and so they're linked to your biological sex because your genotype and phenotype is going to depend on which biological sex you are, because that's going to be determined by whether you have two X chromosomes or an X and a Y chromosome. And so that's going to throw up some specific patterns. And these are kind of the common patterns to look out for that are normally kind of explanations for why certain genes are going to be inherited in a certain way if they are sex-linked. So most sex-linked genes are found on the X chromosome, and so we refer to them as X-linked genes. And this is because the Y chromosome is smaller, it's about half the size, so it carries fewer genes. So Y-linked genes, specific Y-linked genes that we know about, are quite rare. So males will only ever be able to have one allele of each sex-linked gene because they only have one of each chromosome, they don't have a pair. And so they will always express the characteristics of that gene, even if it's recessive. So it doesn't matter what alleles they have on the X and Y chromosomes, they will express them because they aren't a pair, so they won't carry the same genes. They won't have two copies, they won't have a dominant and a recessive, they will just have one recessive copy if they have it, or one dominant copy if they have it. So this means they're more likely to have recessive phenotypes because they can't be carriers of X-linked genes. So any gene on the X chromosome that they have, if they have it recessive, they won't have a dominant copy to kind of mask that, so they will have to have that recessive disorder. And so they're more likely to have any recessive phenotypes that are caused by recessive genes on the X chromosome. Females on the opposite side then, so literally the opposite to that, they get two copies of every X-linked gene. So they're less likely to have recessive phenotypes because they are able to have dominant genes and be heterozygous. And so it's for them, in order to have recessive disorders of genes that are on the X chromosome, they would need to have the two recessive copies, which is obviously, we know, tends to be rarer in the population. They obviously can then also be carriers of X-linked recessive genes, so they can be able to pass those on to their offspring but not have the disorder, whereas males will be able to pass on those genes to offspring, but they will always have the disorder. They can't be a carrier because they can never be heterozygous for any sex-linked genes because they only have one of each chromosome and not a pair. In males, all Y chromosomes are inherited from sperm, obviously, and all X chromosomes are inherited from eggs. So all X-linked genes, including disorders, will be passed on from mother to son. So that's why normally we see these disorders being linked and sort of displaying in males, but they come or have been donated to them from their mothers. Okay, so let's look at an example of an X-linked disorder that we've been talking about. So haemophilia is a good example. It's one of the ones that has gone through, for example, the royal family in the UK. You can go and look this up and you can kind of trace that through the royal family for generations. So it's caused by a faulty recessive X-linked gene, which results in slow blood clotting due to a faulty protein being made. So you get bruised really easily, you can have dangerous internal bleeding and things like that. Heterozygous females, so females that have both a working copy of the gene and a recessive faulty copy of the gene, will be able to pass it on to their male offspring, and they will always be affected because, again, they cannot be carriers. So a carrier female can have offspring with an unaffected male. They are able to produce at least one affected male, and they can produce an unaffected male. They can also produce an unaffected daughter, so a daughter that isn't even carrying, and then also a carrier daughter. So this is one of those examples of a 1:1:1:1 ratio. So 4 different genotypes possible from this cross. But they are able to produce an affected male because you'll see they obviously only get one X chromosome and they'll get the one that has the affected recessive gene. Way we do this, and these Punnett squares are very similar to how we did the Punnett squares for codominance. You just obviously have to remember that you can only use the letters in the boxes. So there'll be no gene on the Y because it's an X-linked gene. It's only found on the X chromosome, so the Y just has nothing. Sometimes you might see them put 0, but that can be confusing, especially if you think about codominance. We could have a capital O, for example. So just leave it blank and just do it in the same way you would do a Punnett square for codominance. So you have your X and your Y, and the X's and Y's are always going to look the same in every Punnett square for doing sex determination, and then you just put the genes that are linked to those sex chromosomes as superscript on the top. So we have our affected male who has the X with the recessive gene H for haemophilia, and then we have our carrier daughter, and she has one of each. So she has an affected haemophilia gene on one X chromosome, and then the dominant copy on her other X chromosome. So she is obviously going to be the same as her mum. She'll be a carrier, and she can then pass this down to her male offspring as well. And then we have 2 children who are unaffected. So if a male gets given the X chromosome has the working copy of the gene, they will be fine. And if we get a female that gets 2 copies, one from male and one from female, which both have a working copy of the gene, then her XX chromosomes will both be fine. A different scenario then is if we have an affected male this time and an unaffected female. Affected males can pass on their affected gene to their female offspring. They obviously can't pass on to their male offspring because the male X chromosomes are only ever coming from eggs, not sperm, because in order to be a male you need to have a Y chromosome. Only sperm can donate Y chromosomes to the gametes. So affected male, he has the recessive gene on his X chromosome, so he has haemophilia and he's reproducing with an unaffected female. So we're going to get a daughter that is a carrier, or 2 daughters that are carriers because they both receive the affected X chromosome from their dad, because they will only get the X chromosome that he has, and he only has one to give when he has the affected gene. And then both males will be fine because obviously they are inheriting a working copy of the gene from their mum's X chromosomes because both of hers are fine. So this is just an example, but you could do this in exactly the same way for any X-linked disorder. If it gives you an example in a question, in an exam question, it will tell you what the genes are, which one's recessive, which one's dominant, and if it's X-linked or Y-linked, and then this is how you would do it in exactly the same way. Okay, so moving on to autosomal linkage. Autosome, it sounds like a fancy word, but it basically just means non-sex chromosomes. So the other 22 pairs of chromosomes in humans are all autosomes because they are not sex chromosomes. So most genes that are on separate or different chromosomes are assorted independently. So the allele received for one gene doesn't affect whether you get the allele for the other gene or not. And we'll look quickly and recap independent assortment in a second. This gives us the typical 1:1:1:1 or 9:3:3:1 ratios that we'd expect for dihybrid crosses between different genotypes of parents. So I've got my chromosomes here. I've got 2 different chromosomes. They're not a homologous pair. And on this chromosome, we have gene 1, and it's not linked to the other genes. So it's not going to determine if you get gene 1, it's not going to have any effect on whether you get gene 2, gene 3, or gene 4, because they're on completely different chromosomes. However, gene 2, gene 3, and gene 4 are all on the same chromosome. So we say that these genes are linked because they are close together on the same chromosome. So they are very likely to be inherited together. So having gene 2 means you'll have gene 3 and gene 4. Like all of those genes are coming together as a package because they are on that same chromosome and they're very close to each other. Because they're very close to each other, they're on the same chromosome, they're going to stay together during independent assortment. They will likely then enter the gamete together because they're on the same chromosome. So if this gamete gets sorted into the nucleus of one of the new gametes, then all of those genes will be coming together. And only crossing over would be able to separate them, because that's the only way genes would be separated from the same chromosome and put onto another chromosome. So we talked about it before, how the chiasma forms when they cross over, the chromosomes break apart, and then literally whole chunks of chromosomes swap with each other. That's the only way we could separate these 3 genes. Now, this produces an unexpected ratio because there's more gametes which contain the linked genes and fewer where we get a random combination of them, or recombinant where they've been swapped over. So this is how normal independent segregation happens. So I've got 4 chromosomes, I've got A and B and A and B genes on my chromosomes, and they get independently assorted into my 4 gametes at the end of meiosis, and we get the usual combinations we would expect. So we get capital A, capital B, capital A, little b, little a, capital B, and little a, little b. This makes sense, and they've kind of not really affected how the other one is inherited. So they will have been separated and pulled apart. They are on individual separate different chromosomes, so they're not linked, and they're being therefore placed into the gametes in a ratio that we would expect, which is 25% of each. So 25% of each gamete would have these gene combinations. Now, if we look at a similar example with linked genes, and I've just halved the number of chromosomes here to make it slightly easier to see. So I've got 2 chromosomes, but they have linked alleles and linked genes this time. So I've got capital A and capital B on one chromosome, little a, little b on one chromosome. Now, it's more likely that little a and little b will travel together on that same chromosome into one gamete, and the capital A and capital B will travel to one chromosome, on one chromosome to a gamete. It's much less likely for crossing over to happen and to have split those 2 up, so we get a different combination where we would get a big A and a little b, and a big B and a little a together. It can happen, and it can happen through crossing over, which we'll look at in a second, but it's just much less likely. So instead of the nice 25% chance of each of those genes being inherited because they're on different chromosomes, because they're on the same chromosome, we're more likely to inherit the same chromosome structure with the same genes as parents, because they will stay on that same chromosome and just move into a gamete when they get separated, than crossing over to happen and for us to get a different combination, or what we call a recombinant chromosome. So if we look at the actual percentages of what we actually get, we've got 48% chance, or nearly 48% chance, of getting the A and B capital letters and the little a and the little b small letters, because they're more likely to stay together and therefore more likely to end in the gamete together because they're on the same chromosome. There's only 2% chance probability that we will get a recombinant chromosome in our gamete because it relies on crossing over to happen, and we'll show you why that is rarer on the next slide.
Okay, so the recombinant gametes, so remember this is where genes that are normally inherited together on the same chromosome get swapped. So instead of the normal 2 alleles that are on the same chromosome that you've got from the parent, then they get switched around, and so we get a different combination of those genes. So that happens through crossing over. So having recombinant gametes with linked genes is evidence that crossing over has occurred. So if you end up getting those combinations of alleles, those combinations of genes that wouldn't have normally been inherited from parents, or they're different from the parental chromosome, then crossing over must have happened in order to have mixed them up. And this happens really often when genes are far apart on the same chromosome, because crossing over happens quite easily and happens regularly between chromosomes. And if the genes are far apart on the chromosome, then they're more likely to end up on different chromosomes, because crossing over is likely to happen in between the gap between those two genes. But if your genes are really close together, if the loci of the gene is really close together on the same chromosome, there's a much lower chance that actually a chiasma, a breakpoint, is going to form between that tiny gap in between those two genes. And so it's just much rarer for those linked genes that are really close together on the chromosome to actually be separated. And so you can see here, I've got my A's and my B's that we had before, that thin green strip is that really short distance between the genes. So it's a very small target area for there to have been an overlap and a chiasma form that would separate them. It does happen. So you can see I've now got my little a and my big B on one chromosome, and I've got my big A and a little b on the other chromosome, which is obviously different to the parental chromosomes, where the capital letters are all together and the lowercase letters are all together. So it does happen, but it's just much, much rarer. So if you do see this mixed combination and gametes with a mixed combination, then that means crossing over has happened. But obviously that's why we get, so previously we looked at, is about 2% of gametes actually have that happen. It's also really helpful because it means we can work out the location of genes on a chromosome. So if you know that these genes regularly get separated from crossing over, the chances are that those genes are quite far apart on the chromosome. If you know that these combinations of genes very rarely come up, so the little a, the big B, the big A, and the little b, you know that the big A, big B genes and the little a, little b genes are very, very close together on a chromosome. So it can kind of help us to map out where these loci are on the chromosomes as well. Okay, so thinking about exam questions then, what sort of evidence are we looking out for? So in questions, you'll be asked to maybe identify what type of linkage or if linkage has happened, and you'll be given obviously various information to work that out, but what is the kind of clues that we're looking for? So evidence of sex linkage, if you get a ratio or you get a number of offspring, there's only going to be male or female offspring with a particular characteristic or a particular combination of characteristics. So that's likely what you'll see, is you'll see that this kind of pairs of characteristics are found only in males or only in females, and that suggests to you that they are sex linked. And then obviously we said all of those other things and patterns we looked at with the sex linked disorders, obviously it only can be transferred on an X chromosome, that suggests it's X linked and things like that. And so sex linked is a little bit easier because it'll be made very obvious in the question if they're talking about X and Y or other sex chromosomes and talking about other characteristics as well. For autosomal linkage, you're going to be looking at ratios. So ratios are going to be much higher for two of the combinations of alleles together. So instead of the normal 9 to 3 to 3 to 1 ratio from two heterozygous parents, you'll see a 3 to 1 ratio, because more offspring is going to have the same phenotype as their parents. So you'll be more likely to get more offspring that will look and have the same phenotype as their parents, the heterozygous phenotype, than you will, say, a homozygous recessive phenotype. Or you're going to get a ratio from a heterozygous and a homozygous parent, which won't be the 1 to 1 to 1 to 1. This is the one that you can kind of normally get as an example, and it's because obviously it's a little bit sort of easier to demonstrate, and also it's not that expected 9 to 3 to 3 to 1, so it can trip you up sometimes. So if we had a heterozygous cross between, we've got a tall plant, short plant, normal leaf, mosaic leaf alleles here. So if we have a heterozygous parent, so they're heterozygous for both genes, so they are tall with normal leaves, but they carry the other two recessive alleles. And then we have a homozygous recessive parent who is short with the mosaic leaves. We cross them, we would be expected to get a 1 to 1 to 1 to 1 ratio of phenotypes. So 4 completely different phenotypes, all of the mixes that we can get. And so we do a Punnett square to show that that's what we'd expect, because they'll ask you what's the expected ratio, use a Punnett square to show what the expected ratio of this cross would be. So that's what we're going to do. We'll demonstrate that it should be 1 to 1 to 1 to 1. And then the question will give you what the actual outcome was of this cross. So there we go. I completed my Punnett square. I'm trying to show you kind of where the T's big T's, little t's and stuff is coming from, so you can kind of see how these different alleles were inherited, because it's make a bit more sense then when we talk about what's actually happened. So I've got my 1 to 1 to 1 to 1. You can see literally I've got 4 columns of the same genotypes. So each column represents kind of the same genotype happening. So I've got a ratio of 1 to 1 to 1 to 1, the different phenotypes. But then I would be given a table maybe with some data, or we'd just be told in the question that the actual ratio was 8 to 1 to 1 to 8. And so immediately you know that something's happening here because it's not the expected ratio, so something must have caused this. Now, because we've got way more of 2 different genotypes and way less, or the expected amount of 2 other genotypes, we need to be thinking about, okay, this must be linkage. Because what it means is we've got way more of the phenotypes in the population that are the same as the parents. So remember, our parents were heterozygous and homozygous recessive. So then you look at the data of what the actual ratio was, and I've got way more of those 2 phenotypes in my offspring ratio. And so they're a lot more common, and clearly that means that they are the parental genotypes, and that's why they're being inherited, because those genes are linked, as opposed to the recombinant ones where we would have had to have swapped those bits of chromosome round through to with crossing over in order to make those different combinations. So more big T, big M gametes and more little t, little m gametes were made because these genes must be linked. So little t, little m must be linked and they must be on the chromosome together. Big T, big M must be on a chromosome together and close together. And so that then is going to give us our linkage, and those are more likely to be inherited together, which is why we're more likely to get a heterozygous phenotype and then a homozygous phenotype from this cross of these 2 parents. Okay, so that's the explanation we need to give. And then the other part of the explanation, what other evidence can we get from this ratio, the 8-1-1-8, is that we still have some recombinant gametes being made, because otherwise we wouldn't have that 1 and 1 with the other 2 phenotypes. So that must mean that crossing over has happened. So we've got evidence of which genes are linked and why, and that's because we've got more offspring, or it's more likely to have offspring that are the same genotypes as the parents, which tells us that the big T and the big M alleles and the little t and the little m alleles must be on the same chromosome and they are linked. And then we've got the fact that we've got some recombinant gametes being made, so some recombinant chromosomes, some mixing is happening through crossing over, and so that's evidence that crossing over must have happened. So that's another bit of evidence we can say. Finally, the next sort of development of these questions, so we've sort of seen lots of different examples of questions that they can ask. They can include the chi-square test in questions like this. So because we know what expected ratios we would be expecting to see for certain genetic crosses, we can use the chi-square test to check if there's significant differences between the ratios we'd expect to see and the ratios that we actually observe in nature. And so then if there is a significant difference, then that gives us evidence to suggest that linkage or epistasis or something else is likely to have occurred. And so that's kind of how they would take this question for you and kind of develop it. So let's use the same example we were just looking at. Obviously, we know that there is going to be a difference because we know what the ratio was and it was different, but this is how they could get you to explain that or demonstrate that. So we did our Punnett square. We said the expected ratio would be 1 to 1 to 1 to 1 with this cross. Then they give us the data of the phenotypes and the actual number of offspring that was produced. So in order to get the ratio, we divide all of those by the smallest number. So we divide them all by 148. And so that gives us the 8-1-1-8 ratio. So we've got our expected ratio and we've got our observed ratio. We can use our expected ratio to then work out what the numbers we would expect to see. So that's obviously going to be effectively 25% of the total for each phenotype, because that would be our 1 to 1 to 1 to 1 ratio. And then we can work out our actual chi-squared. So because this is a statistical test, we have to have a null hypothesis. They might ask you to suggest what the null hypothesis would be for this test. It's, remember, null hypothesis, null meaning no or nothing. So it's always that we're saying there's no significant difference between, and in this case, it's the observed and the expected ratios for the phenotypes that we've got. So we wouldn't expect to see any difference in the number of offspring because we're looking at a 1-to-1 to 1-to-1 ratio, we'd expect the same numbers of offspring for all phenotypes if that was what was following the expected ratio. So we're going to obviously then try to prove that that is different using chi-squared, and then we could potentially go on to explain how this has happened and how it has been different from the expected using the explanation that we've just looked at before. So we're going to put these into a table to be able to work out our chi-squared. Now, you won't have to calculate chi-squared in the exam ever, but you might be given a table to partially fill in. You should kind of just be aware of how we're working out these numbers, because you might be asked to interpret some numbers or fill in a table, and you'll definitely be asked to interpret the result. So here's the table, briefly, just written out. So we've got my expected number, as I said, we're going to divide the total, which was 2,700, the total number of offspring, by 4. So that gives us the expected ratio of 675 for each of the phenotypes, because it would be the same for each one. And then I've put in my number of offspring from the table that I've been given, which is my observed numbers. And then I'm just going to follow through with the calculations. So we need to do the observed minus the expected. And then we'll do the observed minus the expected squared divided by the expected. And then we'll sum that all up together, and that will give us our chi-squared value. Okay, so normally now you'd take the chi-squared value and you'd go look it up into a table. And then if the chi-squared value is higher than the value in the table, then you can say that you are going to reject your null hypothesis. Most likely in the exam, you won't be given that, but you'll be given a p-value, which again, you can work out from those numbers in the table. Or if you do the calculation on a computer, it will give you a p-value. So say I've looked this up and our p-value is actually less than 0.0001. And this is clearly much less than 0.05, which is what we're looking for. We're looking for our p-value to be less than 0.05 for our result to be significant. So with that number and with that comparison, we can now confidently say that we should reject our null hypothesis, because there's less than 5% probability that the difference between the observed and the expected ratios is due to chance. Remember, this language is very specific, and that's the kind of language you need to be writing in the exam. So if you ever get any statistics question, you get given a p-value. As long as that p-value is less than 0.05, that means there is a significant difference. And what that means is that 0.05 means that it's less than 5% probability that the differences that you saw were due to chance. And therefore you can confidently say that it's being caused by something else. And in this case, it's linkage. And we understand that it's linkage, and we explained why in the previous slide. So we've got our explanation of why it happened, and this is just kind of proving that it is linkage. This was clearly very, very different. The 8118 was very, very different from the expected ratio, so it's very clear that there is a difference. It might be in the exam you get a slightly sort of less clear difference, and so that's why they suggest you do the test to see. And then you can obviously go on to explain it in the same way that we just did on the previous slide, and explain that this is linkage and that these genes are linked and therefore more likely to produce phenotypes that are the same as the parents. Okay, so that was linkage, the two different types of linkage. Chi-squared can be asked of you for any question, but it's likely to be asked of you to sort of demonstrate that there is a difference from the observed and expected ratio, and therefore you can then explain it using your knowledge, and they'll want you to say, oh, this is linkage, oh, this is epistasis, because. And so that's why I've put it in here, because it's common to be asked that question in this topic with these questions.
Ouch! This is why in some videos I've explained scratches.